I ended last time by telling you that long after the details of glycolysis and the Krebs cycle and the electron transport chain had been more or less completely worked out, scientists still struggled to understand how the operation of the electron transport chain is coupled to the production of ATP. They understood the mechanism of substrate-level phosphorylation, which is how ATP is produced directly by some of the reactions associated with glycolysis in the Krebs cycle. As we've discussed, substrate-level phosphorylation involves the action of a coupling enzyme, an enzyme that couples the release of energy from an exergonic reaction to the phosphorylation of ADP to produce ATP. But biologists couldn't find any coupling enzymes associated with the electron transport chain. Now, in spite of this failure to identify a missing coupling enzyme, they nonetheless assumed that it had to exist somehow. Biochemists were simply convinced that the enzyme had to be there, because they reasoned this was the only way possible to couple the energy released by a reaction to the production of ATP. There had to be a coupling enzyme in there somewhere. In fact, in some summaries of cellular respiration written at the time, this missing enzyme was even given a name, drawn into drawings. It was called enzyme X. In fact, when I went back and looked at my own notes from the introductory biology course I took in college decades ago, I found that I had been taught about this mysterious enzyme X when I first learned about cellular respiration. Well, as it turns out, I was misled, as was a generation of scientists for many years. There is no enzyme X, and coupling enzymes play no role in the way electron transport powers the production of ATP. Instead, the process of oxidative phosphorylation produces ATP in a completely novel way, one not suspected by those biologists who were so determined to find a coupling enzyme. How the process of oxidative phosphorylation works will be the subject of today's lecture. As I told you last time, oxidative phosphorylation proves to be where the real energy payoff of cellular respiration occurs. That is, it's through this process that the cell converts most of the energy it obtains by metabolically burning glucose into the usable form of ATP. Actually, the solution to the problem of how the electron transport chain could drive ATP synthesis was proposed long before I went to college. In 1961, a British biochemist named Peter Mitchell proposed a radical idea for how this could work. Mitchell suggested that the energy released by the redox reactions of the electron transport chain weren't directly coupled to the synthesis of ATP at all. Instead, Mitchell proposed that the chemical energy released by these redox reactions is converted to a different form of energy, and that it's this alternative form of energy that's used to drive the synthesis of ATP. Specifically, Mitchell suggested that the energy released by the electron transport chain is used to pump hydrogen ions across a biological membrane against their concentration gradient. This active pumping of hydrogen ions, and remember, hydrogen ions are just basically protons, right? They're hydrogens that have lost their electrons, so they're a naked proton. This pumping of protons or hydrogen ions creates a strong concentration gradient. And remember, concentration gradients represent a high energy state, a form of stored energy. The energy associated with this hydrogen ion concentration gradient then is used, Mitchell suggested, to phosphorylate ADP, producing more ATP. Thus, Mitchell's proposal was that the connection between the electron transport chain and ATP synthesis is indirect, with the chemical energy released by the redox reactions of the electron transport chain being converted into energy associated with a concentration gradient before it goes into making ATP. Mitchell's hypothesis came to be known as the chemiosmotic theory of ATP production. This is called the chemiosmotic theory because it suggests that the production of ATP is driven by a diffusion force similar to osmosis. We now know that Mitchell's chemiosmotic theory correctly describes the basic mechanism underlying what we've referred to as oxidative phosphorylation. For many years, however, Mitchell's ideas were rejected out of hand by his scientific colleagues, or at least most of them. People were too committed to their assumption that the process must involve substrate-level phosphorylation and that a coupling enzyme must be involved. so they simply couldn't appreciate Mitchell's way of thinking about the problem. Eventually, however, biochemists began to verify experimentally several key predictions of Mitchell's chemiosmotic theory. And finally, in 1978, in recognition of his idea, Mitchell was awarded the Nobel Prize. Well, let's see if we can understand how chemiosmosis, or the chemiosmotic theory, works. And to do so, it's useful to first step back and remind ourselves where the various processes of cellular respiration are taking place. The key thing to keep in mind here is that glycolysis, the Krebs cycle, and electron transport occur in different parts of the cell. And where they occur, especially where electron transport occurs, is important for understanding how these processes function. Now, glycolysis is thought to be the evolutionarily oldest biochemical pathway associated with the processing of energy by cells, having evolved in some of the very earliest prokaryotic organisms that ever existed on the planet. It's not surprising, then, that glycolysis occurs in the cytoplasm of cells, because that's what prokaryotes have. That is, the enzymes and the substrates and the products associated with the glycolytic pathway are all found throughout the cytoplasm in the cell and in no particular physical relationship to each other. By contrast, the enzymes associated with the Krebs cycle and the compounds that make up the electron transport chain in eukaryotic organisms are isolated in a specialized compartment in eukaryotic cells. A compartment that's been specialized to serve these energy processing pathways in particular. This compartment is the organelle that we've referred to before as the mitochondrion. Now, like all organelles, mitochondria are enclosed by lipid bilayer membranes. But the mitochondrion actually has two membranes, not just one. There's a smooth outer membrane that defines the boundary of the organelle. But there's also a much more convoluted inner membrane. This inner membrane folds back and forth on the inside of the mitochondrion, creating a set of in-pocketings that extend into the center of the organelle. We call these in-pocketings cristae. Now, if you sliced a mitochondrion in half down the middle and looked at it essentially in cross-section, these cristae would look like fingers extending into the center of the organelle. Because mitochondria have two membranes, not one, they actually have two distinct compartments on the inside. One compartment is that compartment that's formed by the space that exists between the smooth outer membrane and the convoluted inner membrane. This compartment is called the intermembrane space. That's an easy name to remember. Now, the second compartment is the compartment that's found on the very inside of the inner membrane. This innermost compartment is called the matrix. Now, given this background on the structure of mitochondrion, let's get back to asking where the processes associated with cellular respiration occur. The enzymes associated with the Krebs cycle are located on the inside of the matrix. Now, one or two of these enzymes actually may form a loose association with the inner membrane itself, but by and large, for our purposes, we can think of all the enzymes, substrates, and products involved in the Krebs cycle that we talked about last time as occurring in the fluid that fills this innermost compartment, what we've called the matrix. The molecules associated with the electron transport chain are physically embedded in the inner membrane itself. Most, but not all, but most of these molecules are kinds of proteins. So their association with this membrane is similar to the other kinds of membrane-bound proteins that we've looked at earlier in other contexts in the course. The fact that the electron transport chain is physically located in the inner membrane of the mitochondrion is really critical for understanding how chemiosmosis works. But before we complete that story, we first want to look again quickly at cell membranes in general. Okay, we've done a little bit of this in the past. I want to review some of this material and extend it. Now, recall that the basic structure of a biological cell membrane or a biological membrane is a bilayer that's composed of a kind of lipid molecule called a phospholipid. These molecules are called phospholipids because they have a phosphate group associated with one end, an end that we usually call the head, or the phospholipid head. And then extending from this head are a couple of long carbon chain tails these carbon chain tails are typically made of a kind of lipid known as a fatty acid. Now, the biochemical details here, again, don't matter, but the point I want to make is that these phospholipids naturally arrange themselves in a neat two-layer row, with the phosphate heads, which are attracted to water because they're charged, facing on the outside of this bilayer, and the fatty acid tails extending into the inside. So it is as though there is a membrane that has phosphate heads on one side and the other, with their tails extending inside in between the two. Now, the point I want to make is that the chemical properties of the phospholipids arranged in this bilayer fashion are such that it's very hard for many other kinds of molecules to cross the membrane. This is a good thing, of course, because what cell membranes do is form boundaries between functional compartments, separating the inside of the cell from the outside, or separating the inside of an organelle from the rest of the cell. So it's a good thing that molecules just can't cross a lipid bilayer. In particular, molecules that carry an electric charge, such as ions, can hardly cross the cell membrane at all. This is a point I made earlier in the course when we were talking about the role of ion concentration gradients, particularly gradients of sodium and potassium ions, in the context of the way that neurons establish a resting membrane potential and fire action potentials. Now, we should also remind ourselves of another thing at this point, and that is that the lipid bilayer that makes up a biological membrane typically has many other kinds of molecules associated with it in one way or another. And in particular, it's usually associated with many kinds of proteins that are attached to one side or the other of the bilayer, or in fact that might cross all the way through the membrane. Ion channels, which we encountered earlier again in our discussion of neurons, are a particularly important kind of transmembrane protein for us to consider again in the context of the way oxidative phosphorylation works. Although charged ions can't cross lipid bilayers, they can cross cell membranes by traversing protein channels that essentially form tiny holes, allowing them to pass. Remember, these protein channels, these ion channels, can be very selective for which species of ion they allow to cross. They can also be passive ion channels, meaning that ions will cross through them only running down their concentration gradient. Or ion channels can be active, pumping ions uphill energetically against their concentration gradient, but only at the price of some energy being invested in the process. Now, the final point I want to emphasize here, again by way of review, is that a concentration gradient across a biological membrane is a form of stored energy. This stored energy is what pushes ions to run down their concentration gradients if an ion channel is available for them that allows them to pass the membrane. Like any source of stored energy then, the energy stored in a concentration gradient across a membrane can be used to do some sort of work. Okay. So now, with that background, let's step back and ask what all this talk about membranes and concentration gradients have to do with making ATP. To understand the connection, remember that mitochondria have two membranes and two compartments. Remember, there's that inner membrane space, which is the region between the inner and the outer membranes. And then there's the matrix, which is the region on the inside of the inner membrane. The enzymes responsible for the Krebs cycle are in the matrix, and the compounds involved with electron transport are actually found in the on the inner membrane itself. Peter Mitchell's chemiosmosis theory proposed the following. He suggested that the movement of electrons through the electron transport chain isn't directly associated with ATP production, as I said, but instead Mitchell argued that the energy released by these redox reactions does the work of actively pumping hydrogen ions, protons, from one side of the inner mitochondrial membrane to the other. That is, he argued that some of the proteins involved in electron transport not only serve as electron receptors and donors in redox reactions, but also act as transport molecules that actively transport hydrogen ions up their concentration gradient across this membrane. Active transport requires energy, and Mitchell proposed this energy was obtained directly from the redox reactions. So specifically what Mitchell suggested was that hydrogen ions are pumped from the matrix of the mitochondria, that's the innermost space, into the intermembrane space, yielding a very high concentration of hydrogen ions in the intermembrane itself and a very low concentration of hydrogen ions in the matrix. In other words, a concentration gradient that would drive hydrogen ions from that intermembrane space into the matrix, if there was some way for them to traverse that space, to traverse that membrane. Now, interestingly, with as much work that has been done on electron transport chain and the molecules involved and the chemiosmosis in the more than 40 years since Mitchell originally proposed this hypothesis, it's actually a little surprising to me that it's still not fully clear how this pumping occurs. We know that it does occur, but we're not sure of the exact molecular mechanisms. What is clear is that the electron transfer molecules of the electron transport chain are grouped into several complexes in association with some other proteins that aren't directly involved in the redox reaction. And it appears that for some of these assemblages, the energy that's released by the redox reactions causes hydrogen ions to actually move through the complex from one side of the membrane to the other, from the matrix side into the intermembrane compartment side. Now, in one case, it actually looks like there's a molecule that grabs a hydrogen and shuttles it from one side of the membrane to the other, again, with the energy required to do so coming from the redox reactions of electron transport. In any case, it's clear that the energy obtained from electron transport is used to move hydrogen ions across the membrane and up their concentration gradient. Well, now that we've seen how the electron transport chain is used to establish a concentration gradient of hydrogen ions, we have to ask, what does this have to do with making ATP? We can start to answer this question by going back to the point that the concentration gradient that's been set up represents stored energy. In other words, what has happened is that the active pumping of hydrogen ions across this membrane has converted the chemical energy released by redox reactions into a stored form of energy in the concentration gradient itself. The last part of the puzzle, then, is to ask, how is this energy stored in the concentration gradient used to make ATP by building a bond between ADP and an inorganic phosphate group? Actually, a key experiment that helped to confirm Mitchell's chemiosmotic theory demonstrated directly that the hydrogen ion concentration gradient across the mitochondrial inner membrane does indeed drive the synthesis of ATP. Now, in this experiment, mitochondria that had been isolated from whole cells, that is, mitochondria that had been taken out of the cells in which they normally occurred and kept in a test tube, were shown to change the rate at which they produced ATP depending on the concentration gradient itself. In other words, it's possible to manipulate this concentration gradient, and when that concentration gradient is manipulated, the rate at which ATP is produced changes. Now, the trick behind this experiment actually was the realization that the outer membrane of mitochondria actually will allow hydrogen ions to freely cross it from the outside of the mitochondria into the inside. What this means is that if you take a mitochondrion that's been extracted from a cell, put it in some solution, and change the hydrogen ion concentration of the solution in which it finds itself, the hydrogen ion concentration of the solution will be the same as the concentration of that intermembrane space. And as you increase or decrease that concentration, you measure different rates of ATP production, as you would expect, with an increased difference between the matrix and the intermembrane space, causing a greater production of ATP, and with a decreased concentration gradient, resulting in a decreased rate at which ATP is produced. Well, having demonstrated that this hydrogen ion concentration gradient drives the synthesis of ATP, we have to ask how. What mechanism connects the concentration gradient itself to ATP production? And has been so often the case throughout this course, the answer to this question involves yet another remarkable protein, a protein called ATP synthase. Now, this is not a hard name to remember. It's a protein, an enzyme that synthesizes ATP, ATP synthase. Now, if you look at ATP synthase, if you look at the structure of it, the first image you get is that it looks something like a lollipop. It's a very complex, large protein complex, and it has sort of one part of it that looks like the stick of a lollipop, and this part of the molecule actually extends into the membrane, the inner mitochondrial membrane. And then it has a part of the molecule, a part of the complex that looks kind of like the candy head of the lollipop. And this part of the protein actually is sticking out on the matrix side of this membrane. Now, if you look a little bit more closely at the molecular structure of this protein complex, what you see is that it actually has three parts. One set of protein subunits come together to form what's literally a tube that extends from one side of the membrane to the other. This part of the structure is called the rotor, and the reason it is will be interesting in a second. Now, a second set of subunits come together to form what's now called the knob, and this is the part that looked a little bit like a lollipop head, and this is the part that is sticking in on the matrix side of this membrane. And there's a third part that connects the two. This is the part that's called the rod. The rod actually sticks in to the rotor and attaches to the knob. Now, this is pretty remarkable because the current model for how ATP synthase functions suggests that it produces ATP by the movement of hydrogen ions through the channel formed by the rotor, causing the whole structure literally to rotate. Now, these hydrogen ions are going to be forced through this channel by their concentration gradient. There's a very high concentration on one side of the membrane, a very low concentration on the other side of the membrane. So what happens is the concentration gradient is going to be pushing these hydrogen ions through. It appears that what happens is that the movement of the hydrogen ions causes these structures physically to rotate around. And what is thought to happen is that the rotational energy of this protein complex actually catalyzes the high energy bond that's formed between an inorganic phosphate and a molecule of ADP to produce a molecule of ATP. Now, what I think is even more remarkable is that scientists have been able to actually visualize this rotation. What they've done is attach an actin filament. Remember, actin filaments are those strings of actin molecules that we encountered when we were talking about how muscle cells work, right? So this is a string of little actin molecules all strung together, and you make these little strings and you attach them to the ATP synthase itself. And when you do this, you can actually, with the right kind of imaging technique, see these little threads going around. In fact, they rotate around when hydrogen is being pushed through this structure. They rotate it around at about 50 revolutions per second. And as I said, this rotation is thought to be what actually catalyzes the bond between phosphate, a phosphate group, and an ADP. Now, how it actually does that is something that people are still working on. But the fact that it does that in some way seems to be well established. There's an interesting analogy you might think of when you think about how chemiosmosis and ATP synthase generate ATP. And that is an analogy to pumping water uphill into a reservoir and then using the water pressure that results from that pumping to drive a water wheel. If you pump the water up, then you have to use energy to do so. But once you've done so, you have potential energy stored in that water as it's resisting the force of gravity. If you open up a valve, the water will rush downhill. And you can use the energy released by that rushing water to actually physically turn something like a water wheel, which then can be used to do some other kind of useful work. In the old days, we might use this to turn a stone to grind grain. Nowadays, we might use it to drive a turbine to produce electricity. The analogy of a water wheel to the way ATP synthase works is all the more remarkable since it does appear that the rushing of hydrogen ions through this channel actually causes a rotation of the molecule. Well, that's how oxidative phosphorylation is thought to work. And you can imagine now why people didn't fully appreciate Peter Mitchell's hypothesis when he first suggested it, because it's actually kind of an unusual, if not somewhat surprising mechanism. It's this process, however, that is ultimately what produces most of the ATP that's generated from the oxidation of glucose. This is called oxidative phosphorylation, by the way, because what ultimately drives it is the oxidation of those electrons that are passed down through the redox reactions of the electron transport chain. So we refer to it as oxidative phosphorylation, and it's the chemiosmotic mechanism that makes this all work. Now, there's one last question I want to ask about the production of ATP about cells, which changes the subject a little bit, but which is to ask how cells can metabolize organic compounds other than glucose. We've talked about cellular respiration over the last few lectures solely in the context of glucose as the starting substrate. But actually, only a few organisms, in fact, probably no organisms, ever really get to use glucose as a substance that they take in their diet. Instead, organisms obtain most of their energy from the breakdown of other kinds of organic compounds, including other complex carbohydrates or proteins or fats. How do compounds other than glucose get used in cellular respiration? Well, in fact, the answer to this question comes from realizing that the metabolic pathways associated with respiration represent a kind of crossroads in metabolism. Remember, we talked about how metabolism is a whole series of interconnecting pathways that connect all of the biochemical reactions of the cell. In fact, the processes associated with glycolysis and the Krebs cycle are at the very center of the metabolic map of the cell, really providing a crossroads for all of these other reactions. Practically, what this means is that respiration can accept a wide range of organic molecules for these catabolic reactions. It's just that these organic molecules have to enter the pathways through different ports, through different doors to get in. A wide range of carbohydrates, for example, these are more complex sugars than glucose, can be broken down and fed into what's really the furnace of glycolysis at different steps along the way. Proteins can also be used for fuel. They have to be broken down into amino acids first, but then these amino acids can be converted into chemical intermediates that are fed right into the Krebs cycle or into the pathways of glycolysis. Fats also feed into the metabolic pathway in the middle of glycolysis or in the production of acetyl coenzyme A directly. My only point here is that although we've illustrated cellular respiration using glucose as a convenient starting point, in reality, the complex crossroads of metabolism can bring almost any kind of organic compound into the process at some point, making it possible for a wide range of organic fuels to contribute to the production of ATP by the cells in an organism. Well, we've covered a lot of ground over the last several lectures, following the fate of the energy originally stored in the carbons of glucose all the way through glycolysis, the Krebs cycle, oxidative phosphorylation, and finally to the production of ATP by the remarkable enzyme ATP synthase. Let's just step back now for a second and review one last time what the cell has gotten out of all of this. After all, what we're interested in is where ATP comes from. If we do all the calculations, if we figure out how much ATP is produced by substrate-level phosphorylation and also by oxidative phosphorylation, which is a little tricky to do because you have to figure out a number of factors that are going to affect the way chemiosmosis works. But if you do these calculations, you come up with a rough estimate that out of the Krebs cycle and oxidative phosphorylation, we end up producing about 34 molecules of ATP from those electrons that we originally captured from the process and put into the electron transport chain. If we add to that those 4 molecules of ATP that we know we've gotten already out of substrate level phosphorylation directly, this gives us a yield of about 38 molecules of ATP produced, about, for every glucose molecule oxidized. So how good is this? Well, I told you earlier that glycolysis only extracts about 3% of the energy from glucose. If we consider all 38 of these molecules of ATP, what we come up with is a figure of about 40% of the energy originally found in glucose, the free energy in glucose, being converted into ATP. Well, you might say 60% of it got away somehow. We only got 40%. But if you compare that to human engineering, it's really pretty good. A very efficient automobile engine, for example, which also is burning organic fuels in a different way, has an efficiency of only about 25%. So really, these cells are quite efficient. There's one final point to make, though. We might think that glycolysis, because it's so inefficient, really isn't good enough to sustain life. But remember that for a long time after life first evolved, glycolysis was the only set of reactions available. Life can exist on glycolysis alone. It did for many years early in evolution, many eons, and it still does today for many organisms. Yet, when the more efficient reactions associated with cellular metabolism of the Krebs cycle and oxidative phosphorylation finally evolved, then organisms could really take off. They finally got a source of energy that would allow them to become bigger, more complex, and multicellular. And with this added complexity, life began to emerge in a much more diverse and wonderful way.